Okay, this is the last question for the 2022 FRQs. This is number 6. So let's just get into it. This one says, researchers are studying the use of RNA vaccines to protect individuals against certain diseases. To develop the vaccines, particular cells are first removed from an individual, then mRNAs coding for specific proteins from a pathogen are introduced into the cell. The altered cells are injected back into the individual, where the cells make the proteins encoded by the introduced mRNAs. The individual then produces an immune response to the proteins that will help to protect the individual from developing a disease if exposed to the pathogen in the future. When introduced into cells, the mRNAs used for vaccines must be stable so they are not degraded before the encoded proteins are produced. Researchers developed several modified caps that they hypothesized might make the introduced mRNAs more stable than mRNAs with the normal GTP cap. To test the effect of the modified caps, the researchers produced mRNAs that differed only in the cap structure: no cap, normal cap, or modified caps 1, 2, and 3. They introduced the same amount of each mRNA to different groups of the cells and measured the amount of time required for half of the mRNAs to degrade, also known as mRNA half-life, and the total amount of protein translated. Okay. So you don't really need to know too much about the background of RNA vaccines. All this is really saying is they're taking cells out and they're injecting mRNA into them. They don't want that mRNA degraded because they obviously want that mRNA to produce the proper proteins. Okay, so we know that a big issue is mRNAs getting degraded if they don't go through the post-transcription modifications, right? The cap, the tail, the splicing, all that. So they have figured out that maybe adding a different type of cap might protect it more. Okay, so what they're going to look at is basically all 5 different types of caps and how that affects how fast they're degraded. You don't want, obviously, them to be degraded very fast because you want them to have the opportunity to make the proper protein. Okay, so they measured it with mRNA half-life. So that is just, again, like they said, the amount of time for half the mRNAs to degrade. You obviously don't want them to degrade very fast. And then the total amount of protein translated. Your goal is to make this protein, so you're going to want a high amount of protein translated. So you've got 5 different kind of samples or structures that we're looking at and 2 different measurements. Okay. Alrighty, so this one says, based on the data, identify which cap structure is most likely to protect the end of the mRNAs from degradation. Okay, so what we're looking at now is which out of these 5 are going to give you the least amount of degradation. Now, the best way to look at that is to look at the mRNA half-life. So, in case you've never looked at what half-life is, again, it's the amount of time required for half of the mRNAs to degrade. So you have to think about whether you want that number to be small or you want that number to be high. The amount of time for half of it to degrade. You would want that number to be quite high, right? So what this no cap one is saying is that in 1.4 hours, half of the DNA or half the mRNA is degraded. Not great. In 16.1 hours, half of the mRNA is degraded for the normal GTP cap. Okay, so in this case, what we're going to be looking for is the one that has the highest number, right? Because that means it took the longest to degrade the mRNA. So in this case, the answer for this one is modified cap 2. You could also go based off the, not as great of a measurement, but the total amount of protein translated, because that's going to be directly affected by obviously the amount of mRNA you have. But either way, modified cap 2 is highest in both of them. You want these numbers to be high because it indicates that they're going to be around for a long time and they're also going to make a lot of protein. A just says identify, so you don't have to justify it or anything. It's asking you which cap structure protects the mRNA from degradation. This is the most protective because it takes the longest for that mRNA to degrade. So we'll put modified cap 2. Okay, that's all you needed for that one. B says, based on the data for the mRNAs with modified cap, describe the relationship between the mRNA half-life and the total amount of protein produced. Okay, so I kind of already gave this one away, but you're kind of looking at the relationship between half-life and amount of protein produced. You don't have to draw a graph for this. That would take too much time. You can visually look at this, but also it makes kind of sense. Like I said, the more mRNA you have, the more protein that you can have, right? You're basically having more of the ingredients to make the final product. But you're gonna wanna look at the data for this one, and just double check with this. So if I look, and I'm looking for a correlation between these, like I said previously, modified Cap2 had the highest half-life and also had the highest total amount of protein. Let's look at the lowest one now. The lowest mRNA half-life also had the lowest amount of protein, okay? The second highest mRNA half-life had the second highest total amount of protein. And I can keep going with this one. The second lowest, 15.5, had the second lowest total amount of protein. So the relationship for this is a positive correlation, that as I increase the half-life, you're also increasing the amount of protein translated, right? That makes logical sense, and it also makes sense with the data. Okay, so you can say, there's a couple different ways you can kind of say this. Let me just give myself a little bit more room here. Okay. So for B, you could say that it's a positive relationship. Because again, as the half-life increased, the amount of protein also increased. You could also just say that the longer the mRNA half-life, the more protein produced. Okay, I'll put that as well. More protein produced. Either one, they mean the same thing. Okay. Yeah, you didn't need to do a whole graph for this one. Obviously, the graph would have given you the answer, but for time's sake, if they don't make you draw a graph, then don't draw a graph. Okay, C says, after examining the data on mRNA half-lives and the amount of protein produced, the researchers hypothesized that each mRNA molecule with modified Cap1 was translated more frequently than was each mRNA molecule with the normal GTP cap. Evaluate their hypothesis by comparing the data in Table 1. Okay, so I'm going to erase this because we're only looking at 2 of them. They hypothesized that mRNA with modified Cap1, so we have this, was translated more frequently than each mRNA with the normal GTP cap. So they're not asking you if it's true or not, they're asking you to justify it. So let's look at these numbers. So modified Cap1 has an mRNA half-life of 15.5, and its amount of protein is 4.7. The normal GTP cap is 16.1, and the amount of protein is 1. So if we think about this, these numbers are not very far away from each other. You've got 16.5, and then you've got, 15.5 and 16.1. With their standard errors, they actually overlap as well, so they're not really different from each other. However, if we look at the amount of protein that's been translated, there is almost 5 times as much protein for modified Cap1, which is going to justify our hypothesis, which says that for the amount of mRNA, it's translated much more frequently with modified Cap1. Hence why with only 15.5 hours for half-life, you get almost 5 times the amount of protein. Okay, so you're looking at them in relationship to each other. It makes it quite easy because these 2 numbers are very similar to each other and these 2 numbers are very, very far away from each other. You can tell that modified Cap1 was much more translated per starting amount of, or per, half-life of the DNA, or mRNA, sorry. So what I would say for this one is it's supported because the half-lives are similar. For modified Cap1 and normal. Oops. And then I would say, but Modified cap1, I would say produces like 4 times as much protein. So and another thing is they start with the same amount of mRNA, which is important. So if they start with the same amount of mRNA and they have roughly the same half-life, that means they have roughly the same amount of mRNA at any given time. So if we take that, both of these have the same amount of mRNA at any given time, but one of them is, making 5 times more protein, that one's being obviously translated a lot more. 5 times more protein. Okay, so I think that should be good. Yeah, so it's translated more. So it produced 5 times more protein, thus it was obviously translated more. All right, our last one for 6 and for the entire 2022 exam says introduction of mRNAs into cells allows the cells to produce foreign proteins they may not normally produce. Explain why the production of a foreign protein may be more likely from the introduction of mRNA than DNA into cells. So this is a little bit of an application question. Again, we're injecting this mRNA to make protein that's going to help with immune function. Explain why the production of a foreign protein may be more likely from the introduction of mRNA than DNA. So it's really asking you, why are they injecting mRNA but they're not injecting DNA? Okay? So think about that and why mRNA would be easier and also more effective. Okay, so there's a couple different things that you can say for this one, but what it really comes down to, if we look at our central dogma, it goes DNA, mRNA, and then protein, right? This is our goal, right? Our goal is to produce the protein. They're asking you, why are you inserting mRNA instead of DNA? If you really look at it logically, mRNA is further down in the process, right? If I were to inject DNA, I would need it to be transcribed and then translated. But if I just inject mRNA, I only need it to be translated. You're reducing the possibility of error by quite a lot, right? You're skipping transcription. Whatever error can happen from transcription or post-transcription is completely gone if you're injecting mRNA, okay? So that's why you don't want to inject DNA in this case, because then it has to go through transcription and then it has to go through translation. It can have errors in transcription. It can have errors in, the modification process with splicing, the caps, the tails. If I already put a completely modified mRNA in, all I'm relying on is translation. There's just a lot less room for error, okay? So you're just injecting something later that's a little bit later in the process to avoid that error. So protein production from DNA requires transcription. So how am I going to put this? There's a couple different options. Obviously, you only have to say one. You say DNA requires transcription. So that's just an extra step that mRNA doesn't need. Less opportunity for error. You can also say post-transcriptional processing. So DNA also requires modification. So you're also skipping that part, so you're skipping any potential error there. DNA requires processing. mRNA does not require processing because they're already injecting it as the full tail, cap, and everything. And then the last one is just another option. It says that the cell may be unable to transcribe the DNA. Okay, so that's just, again, pretty much an error with transcription, right? Again, if it's not native to the cell, it does bring in issues because, it's not used to having that sequence of DNA. There is a possibility that, it might just not be able to transcribe it. It might not have the proper transcription factors, a whole lot of issues. Really, that comes down to DNA requires transcription. Transcription is an extra step. It's going to require, or it's going to cause a lot more issues than if I were to just inject mRNA. Okay? So that is actually it for question 6, and that's it for the 2022 FRQ answers. If you have any questions, let me know. Thanks.